So this section is actually two. There's section 7B and 7A. So in this section, it's the DNA section, and right, we focus on how DNA is replicated first. So when we replicate DNA, right, we're going to use DNA and make more DNA. So every time a bacteria divides, this needs to happen before that. Right? You need to have 2 copies of the DNA for each bacteria, one for each bacteria. And then when we talk about transcription, the DNA makes RNA. And then translation, we're going to use messenger RNA at the ribosome to make protein. Okay, so a chromosome or genome is all the genetic information for an organism, right? With E. coli, I think it's like 4,288 genes. We have approximately 20,000 genes. You know, when you think about our DNA, right, it's chromosomes and it's linear. Bacteria have a circular piece of DNA, and then they generally draw a couple circles like that, or one. You know, a plasmid is separate from the chromosome in bacteria. So we'll talk a little bit about plasmids too. So this would be a model of the genome of E. coli. It's circular, so they use a circle to denote it. Right? And like this would be a million base pair, 1.1 million. This would be 1.3 million, 3.4 million. That's what kill the base pairs. So if they have over 4 million bases. Right? So what is a base? Right? We say they're little d NTPs, which are nucleotide triphosphates. So there's little d ATP, little d GTP, little d CTP, little d TTP. So we're going to put these nucleotides together, A, G, C, or T, right, to make a strand of DNA. To make a strand of E. coli's DNA, you need 4 million of these nucleotides. Each one of them represents what they call a base pair, because it's double-stranded. But so there isn't much to know about this figure other than just to illustrate the fact it's circular, it's big, there's 4,288 genes, and that's what makes E. coli E. coli, these specific 4,200 genes. Now, other bacteria share the same genes, right? But what makes E. coli E. coli unique is those 4,288 genes. You call that their genome. If there's genes located on the plasmid, however, they're not really considered part of the genome or E. coli's genetic information, right? It's just an extra piece. Without that plasmid there, it's still E. coli. So DNA, right, it stands for deoxyribonucleic acid. It's double-stranded. It contains nucleotides. Each one of these is a nucleotide, A, G, C, T. Triphosphate means there's 3 phosphates in it. You need these to make DNA. Okay? So, right there, in each nucleotide, they have a sugar, deoxyribose, and DNA. There's 4 different bases that can make up the nitrogen base, and then they all have phosphate groups. Every time you have an A, it hydrogen bonds to a T in the opposite strand. So you tend to show DNA, if you made it a ladder, this is supposed to represent one strand, this represents the other strand. If you don't show the sequence, you may represent bases. This strand is hydrogen bonding to that strand with these lines. If there's an A in this strand, then there's a T in that strand. Right? If there's a G in this strand, then there's a C in that strand. So that's where you get these basing. This strand of DNA is not identical to that strand. We say it's complementary. Right? We talked about that a little bit in the macromolecule section. You know, I don't expect you to understand. It's a difficult concept, especially when you put a twist in it. Right? This is a ladder, let's say, and it's flat, but DNA is helical, so you really have to twist the ladder to, like, it looks like a spiral staircase. But if, you know, this is the, you know, when I look at this, I know what all the little circles are. I wouldn't expect you guys to know it, but that's what it, if you could look at DNA, it would resemble that kind of look. Not those colors, but, so, you know, you can show DNA, you know, like this, you know, you can show it like a spiral staircase. There's no A, they don't tell you what the bases are here. Here they're trying to give you an indication. But each one of these little steps is, that's one base pair, that's one step, that's another base pair, that's the next step. You just, there's over 4 million base pairs, which means these two, right? There's 2 for each pair. This strand is covalently linked. This strand is covalently linked, and the 2 strands hydrogen bond. It's a minor point. We say that, so if we just look at a small part of the strand, right, these 4 bases, they're actually all 4 of them. They hydrogen bond to these 4. That's what you'd expect. It's a small, when we say sequence of DNA, then we're saying we know what this T is, what this one is, what that one is. You know, the next one could be a T. The next one could be a T. The next one could be an A, right? It goes on and on. 4 million. So, this strand is complementary to that strand. It's not identical. It's anti-parallel, meaning there's the 5' ends here, the 5' end of this strand's over there, and that just by labeling that, it helps you see that this strand goes in this direction, and this other strand goes in the opposite direction. So when you see DNA, a sequence of DNA written out, if you did it horizontally, say A, T, A, G, A, you would start from this side and read it in that direction. You know, it's a 50-50 chance. Which way would it go? It just happens to be the same direction that we read English. So in section 7A, we're going to go over DNA replication and gene expression. So gene expression is both the transcription plus translation. When we read a gene, which is in the DNA, right? E. coli has 4,288 of them. They're not always being used. If you want to express the gene, then what you do is you convert the DNA to RNA, and then the ribosome converts the messenger RNA into a protein, and then you express that gene by making the protein. And then in 7B, we'll talk about recombination of mutagenesis, what that means. So when one bacteria becomes 2, you need to generate 2 copies of the DNA, or a copy of it, so that you have 2 copies. Right? We're going to make that, turn that into 2 copies. Right? We start off with 1 copy There's the 4,200 genes, and then we're going to end up with 2. So what happens first? Let's say this is one strand of the DNA, and then here's the other strand. The first thing that happens is you separate the strands. Right? All of this is double-stranded. And then at this point, we've made a bubble, they call it. So what happens this happens it initiates, where does the DNA replication initiate? It starts at the origin of replication. Right, that's a specific spot in the DNA. It opens up there. Then once this is open, DNA polymerase, which is the enzyme, this is the enzyme that uses the NTPs, A, G, C, T, they polymerize. So we make another strand, right? We're going to convert this single strand into double-stranded. And then we're going to convert the top one into double-stranded. Okay, and then right here where they are coming apart, we call that a fork. So fork here, there's a fork there. So what the fork's going to do is keep moving in this direction, and this fork's going to move in that direction. So what they're trying to do in this depiction, right, they're showing it, it started here, and then this fork moved down to it got here. Now it's going to keep moving. This fork's going to do the same thing, right? And you keep going. You turn it into, you open up the two strands, the old strands, and then let's say we move this down a little bit more, right? This fork moves this way, and this fork's moving this way, and then this strand becomes double-stranded in the process, and then this strand, and then the fork's down here now. This strand moves, turning that single into double. It's not that complicated. You don't worry too much about what's actually happening. I just want to sort of explain it. When you go from, you have a single strand piece, if there's an A, C, say G in that strand, that's single, then the nucleotide that hydrogen bonds to A, which is the TTP, that comes in, and you make that. Well, let's say we don't have that. Still single here. Then the nucleotide, right, the G comes in, and then C. So that's what's happening when you're converting the double strand or the single strand into double strand. And don't worry too much about, if you don't quite get that. Here's another depiction of, right, you make the bubble, you've got a fork here, you have a fork here. And then right, so you've already put 3 of them together, then the fourth one comes in. If there's an A here, like I said, if that was an A, then you would bring in a TTP. And then the TTP now this one would be 4 bases long, right? It's 3 now, it's 4. And you look at the next base, that tells you what the next base will be. So you're making a complementary copy of the single strand. But that single strand is dictating what bases go into the new strand. Okay? So I just make it, when I use the term melt, I say that's what we mean by separating the strands, and then DNA polymerase is the enzyme that makes a complementary strand of the single strand of DNA. And in the process, it converts the single strand into double strand. So we refer to it as being a semi-conservative process. There's what's called Ozaki fragments and a leading and lagging strand. Don't worry about that for our purposes. That's not important. So it's pretty complicated. One strand of DNA gets made in one direction, the other strand gets made in the opposite direction, and it gets way complicated, to say the least. Okay? Now, what I did on the board, right, we've got initiation, where it begins. Then we have elongation. What's that process entail? In this case for DNA, it involves this enzyme, DNA polymerase, right? That's where it begins at the origin of replication with DNA polymerase. To get the elongation, the polymerase polymerizes these 4 nucleotides. It makes a newly synthesized strand. So we say the energy for the process is found in all of these, right? Little d NTP refers to all 4 of the nucleotides, whether it's an A, G, C, or T. The difference between the 4 is their nitrogen base. I think I got bit by a flea. It just feels like it. The problem, you get bit once. If you don't kill it, then you get bit again. Now, termination is not a big deal for DNA replication because as soon as the forks meet, it's done, right? There's nothing to test you on, I guess is a nice way to put it. So, DNA is another word for it. Polynucleotide just means it's many nucleotides. So sometimes you see it referred to that way. You know, nucleic acid or polynucleotide means the same thing. So I explained anti-parallel. One strand goes in one strand goes in the other direction. You say DNA is directional. One strand is complementary to the other strand. There's a phosphate backbone. I didn't really talk too much about that, but that's how if one strand is they're all linked together covalently that way. And one strand of DNA is hydrogen bonded to the other strand. So properties of DNA, I might ask you about, or where does DNA replication begin, that kind of thing. So, for instance, you don't want to confuse it with transcription, right? With transcription, it begins at a promoter. And instead of DNA polymerase, we're going to use RNA polymerase. And instead of the DNA nucleotides, we're going to just call them NTPs, right, which are ATP, CTP, GTP, and UTP. Right, these 4 nucleotides are polymerized by RNA polymerase to make RNA. So I put this on the board just to help you see DNA replication versus transcription and translation, because it's easy to mix them up. So, we did the DNA replication part, right? Gene expression means we want to express the gene. So at the end of this section, I'll talk a little bit about how we control gene expression or regulate it, because we don't always want to be making the protein unless it's needed. Right? That's what gene expression is. We refer to the promoter as the on-off switch. Right? It's found in the beginning of genes. So if the promoter is turned on, you get gene expression. If the promoter gets turned off, you don't express the gene. right? So if you have 4,200 genes, you can't express them all at the same time. It just isn't going to work with E. coli. So it's going to express maybe 10 or 20 or so at a time, maybe more. It just depends on what it's doing. I mean, I have no idea myself. It's the same thing with us. We have 20,000 genes. You know, what are we maybe using at most a couple hundred of them at a time? Hard to say.
Sorry, which section are we working out of?
7A.
Okay, perfect, thank you.
Sorry, yeah, you missed that. I'm videotaping it, so what you missed, you can watch that part of the video. Yeah, I'll have to make it or process it or whatever tonight or tomorrow, so it might be a day before I do that. So, to express a gene, we have to first convert the DNA. We say you copy it, which it really isn't exactly a copy, but it's a complementary copy, right? The messenger RNA is what's read by the ribosome. You use tRNA, ribosomal RNA at the ribosome, so that you translate the nucleotides into a protein sequence, a sequence of amino acids. That's what translation. So we'll first get into what transcription is. Oops. Oh, I already did it, just plugged it. Gotta watch where I put my feet over here. It's not that one. So transcription, or what is a gene, right? It's a sequence of nucleotides that make up, right, a functional product, which most of the time is a protein. So we tend to show in DNA, right, we just say, here's the promoter, and then here's the gene. This is made of DNA, the gene is made of DNA. If the promoter's turned on, then the DNA of the gene gets converted to messenger RNA at begin. That's what we're going to do first with transcription. Expressing it, the protein that's made has to be used by the cell technically for it to officially be expression. All right, and then we're going to go through the transcription, then the translation, which is what we mean by that. So RNA is very similar to DNA. You use different nucleotides. They have similarities in that you use the same adenine, cytosine, guanine, but instead of thymine you use uracil. So they have that in common, the three bases. They use a different sugar, ribose instead of deoxyribose. But the nucleotides, right, are what make nucleic acids, whether it's DNA or RNA. Okay. So we say when we make RNA, we use uracil because thymine is like replaced in the RNA with uracil. So what I'll do is, well, you might see a question if this is A, C, G, A, T. If this is the sequence, what's the complementary sequence? Which is, right, there's always a T for the A, G for the C, C for the G, T for the A, vice versa. Right, for when you get an A, hydrogen bonds to a T. So if I said, well, if this is the DNA sequence, what RNA sequence would it generate, then write out the DNA first and then just replace the T's with U's. That's a way that will keep you from missing it. Kind of a tried and true method. So you might get that question on the test. So, these are the three main types of RNA, right? There's messenger RNA. It contains the ribosomes. If you notice, I don't even put a little M in front of RNA here, because it's all three of the RNAs are going to be made in transcription. It's the messenger RNA that really does most of the work, or what we always think of. So I'll explain how that contains the codons. To make the ribosome, you need ribosomal RNA. Transfer RNA is called the interpreter molecule. Each one of these tRNAs is going to have a correct amino acid attached to it. So when it goes to the ribosome, it's going to know what amino acid goes into the protein based on what codon is present at the ribosome at that time. The codon and the anticodon hydrogen bond. So for instance, if the codon at the ribosome is U, then the anticodon found in the tRNA would be A. Right? Because U's hydrogen bond to A's. So you see, that's how they come together based on that base pairing, right? If there was a C in the RNA, then the other RNA would have a G. If there was a G in this one, it would be a C in that one, vice versa. Okay, any questions about that? I'm going to show you some examples in a minute. So what's a codon? We'll get into that later. Let's first go through transcription. So as I said, where does transcription begin? At the promoter. We say the promoter is in front of the gene. You don't want to start copying the gene in the middle. You want to make sure you want to start before the gene actually begins. You don't start right at it. But technically, to make the protein, the RNA needs to have the start codon, right, which is AUG. I'll talk about that in a minute. But that would be the first codon to give you the first amino acid in the protein. So, in order to begin the DNA, just like when we did DNA replication, we had to open up the DNA. It had to become single-stranded. That's what this is supposed to be, this bubble, right? It's double-stranded here, it's double-stranded there. We open up the strands so we can copy one of the strands. Right? So what happens is the nucleotides come in one at a time. Okay, so let's say we've already made this part of the RNA. If there's a G in the DNA, then it would be a C would be the nucleotide that would hydrogen bond to it there, right? We've already made this part, and we want to make the next, add the next nucleotide to it. It knows which nucleotide based on what the sequence is in the DNA. So, if you, just like before, right, you say if this is the DNA, A, C, C, G, if that's the DNA strand, and we want to, we're making RNA, let's say we've already made this part, for the next nucleotide in the RNA to go in, we have to hydrogen bond, what, a U to A. right? A's and U's go together. So that's the next nucleotide in the RNA. If you add a G there, or a C in the DNA, then we bring in GTP and add it to the U, right? The strand would keep growing. We bring in another GTP and bond it to that G. So this strand gets lengthened with RNA polymerase with these nucleotides based on what the DNA sequence is. Now, at some point, we don't want the RNA and the DNA to stay together. The RNA falls off. But you just keep reading, right? The sequence in this is determined by what the sequence was in the DNA. Okay? Based on if there's a D in the, or an A in the DNA, then you put a U in the RNA. We're good? If you don't get that, it's okay. Don't worry. It's complicated, I know. Now what happens is, right, what we're doing is the DNA is copied to RNA. That's the important thing to remember during transcription. Okay? Elongation is RNA polymerase. That's the enzyme that's used for transcription to make RNA, polymerizing these nucleotides into a strand, one long strand. It stops when it gets to what's called the poly A. So termination for transcription is poly A. You call it a region. And that just means it encounters a bunch of A's in the DNA. A bunch of A's together. Some U's, but mostly A. That's what ends transcription, right? Determination. So just as we did with DNA replication, right? You need to know where transcription begins, what it refers to. DNA is made into RNA. RNA polymerase is the enzyme. The nucleoside triphosphates are the NTPs that I wrote on the board, but I erased them. They're polymerized with the polymerase. Okay, the promoter is referred to like as an on-off switch where this starts, and poly A is where it terminates. And you've made the RNA. So let's say we made messenger RNA. The messenger RNA now is going to contain the codes so that the ribosome can make the protein. Okay, so that's basically the first part of gene expression is transcription. That's where you generate the messenger RNA. You also need ribosomal RNA and tRNAs. Now I'm going to go into the process of the translation of the messenger RNA, right, into, right, if you think, what is protein? A protein are a chain of amino acids. Right, just like RNA is a chain or polymer of nucleotides.
Could you call the protein polypeptides?
Yeah, sometimes polypeptide, yeah, is another name for protein. Peptide is sometimes given the short protein.
Okay.
You don't need to know the distinctions, but yeah.
Okay. I'm just asking because I put polypeptide on the assignment paperwork.
Yeah, that wouldn't work it wrong.
Okay.
No, that's not a problem. Yeah, when I'm grading the assignments, you know, I'm looking for how much time you put into it. If I see it looks like, you know, I see a couple words for an answer, and it might be the right answer, and I might not deduct anything, but I'm kind of looking at that, you know. The more information you put in there, the better. I graded the P and Ps today, and I think some people didn't do the first couple, and I don't think they watched the video, or they're not coming to class and they didn't listen to my explanations. It's you can tell it's real easy. But they'll have an opportunity to redo them. All right, so the genetic code is a list of all the, let's just go to that first, right? Here they're all listed. So RNA contains a U, a C, an A, or a G. The code is 3 of them together, right? So you have 3. This is the first one. So all of these in this row contain, the first letter is a G, the first letter is an A, C. In this column, the second letter is a U, the second letter is a C, second letter is an A. Then the third letter can be all 4 of them, U, C, A, G, right? So at the end, they're kind of written in order. So this is a systematic way of showing all 64 different variations of these 4 nucleotides arranged in 3s. So if the codon begin, as GUU, that tells the ribosome valine goes in the protein. The start codon is AUG. So AUG is always the codon that is at the very beginning of all protein, and you always get that amino acid for the first amino acid in proteins. Then the second codon would tell you what the second amino acid is. So if you had AG after that codon, then you would put glutamic acid as the second amino acid. So that's what the codons instruct the ribosome. So we'll go through that process in a minute. So you find the genetic codes in the messenger RNA. It's universal, unambiguous. It's redundant because we have 4 codons for 1 amino acid. Right, that's what we want to do. Right, we want to use the genetic information, AUG, to tell the ribosome which amino acid is going to be in the protein. So if we have 100 amino acids in a protein, we need 100 codons. In other words, right? A codon for each amino acid. There's 3 stop codons, right? That means if you get to, you see these 3 codons, then, right, UAA, UAG, and UGA, then you've finished making the protein. So if it's 100 amino acids, you'd have 100 codons, and then the 101st codon would be one of these 3. So you have 20 amino acids, and you have 61 total codons for amino acids. So you have more codons than you do amino acids. That's why you end up with, now some codons, like tryptophan only has 1, but most of them have at least 2, if not 4, and I think there's 1 with 6. Leucine actually has 6. That probably means leucine is used more often in proteins than the others, would be my guess. It probably depends on the organism, but so that's just the list of the codons. So just to recap now what we've already learned, right? Transcription means we're going to convert one of the strands of DNA into RNA. There's 3 nucleotides. They don't tell you what the 3 nucleotides are. But for instance, we know AUG is always the first. This would be an A, this would be a U, this would be a G. That's the information to put in the first amino acid, which would be methionine, which I said is always the first. Then the next 3, let's say it was UGG, were the next 3 nucleotides. Then we know we put tryptophan would be the second amino acid. right? If we went to the third amino acid, and let's say it was GUA, then we know valine would be the third amino acid. So I'm really getting into the nitty-gritty details of this. You know, when I ask the questions on the exam, it won't be so detailed. And just like this, if you don't quite get this, don't worry about it. So at the ribosome, when say transcription or translation begins, we're going to start with this codon. That means the RNA, the transfer RNA, will have a UAC, which is what is complementary to that codon, right? A to U, U to A, G to C. So that's why this tRNA goes to the ribosome and brings in methionine. If the second codon is CUG, then the transfer RNA with GAC would come in. There's 61 of these. There's one. They differ, right? The anticodons, there has to be an anticodon for each one of these codons. So that when the transfer RNA with the anticodon comes to the ribosome, it brings the correct amino acid. So the other thing you have to assume is that if the transfer RNA has this anticodon, it always has leucine attached to it. If it has this anticodon, then it has to have threonine attached to it. The transfer RNA transfers the amino acid that's supposed to go in the protein to the ribosome. At the ribosome, the amino acids get linked together, one at a time. Right, so the translation initiation complex, that's what's formed to initiate translation, right? It's the initiation, it's called the initiation complex. So it includes ribosomal RNA in the ribosome. It includes the messenger RNA for whatever protein we're going to make. And then the first tRNA is always the one for methionine. So methionine, it doesn't show it attached, but before it gets to the ribosome, it needs to have the correct amino acid attached to it. Now once that occurs, then you bring in the second transfer RNA, and that's dictated by what the codon is. If this is the codon, that gives you that anticodon. So this is a codon for leucine. So leucine would be attached to the transfer RNA. So if you've never learned this before, you know, it's a head scratcher. So once you bring these two to close proximity at the ribosome, then they get linked. You form a peptide bond between them. And you started making the protein. So the next thing that happens, right, this transfer RNA, number one, it leaves its amino acid in the protein, but then it leaves the ribosome. If it comes back to the ribosome later, it has to have the transfer RNA attached to it. So there's a process where that happens. And then you move down to the next codon in the messenger RNA. The ribosome does. And then because of the codon and the anticodon, the third transfer RNA attach. It brings in proline, right? Because the codon, this codon is for that amino acid. You bring the two amino acids in close proximity and you generate a peptide bond between amino acid 2 and 3. The leucine and the proline. So you just keep doing that. You keep reading, the ribosome keeps reading the messenger RNA. That dictates what amino acid goes into the protein at the specific spot. There's like you number these amino acids, right? You have amino acid number 1, there's amino acid number 2, there's amino acid number 3, amino acid number 4. They have to be correct. If they're not correct, then what do you call that? It's a mutation, right? A protein, a mutation in the DNA can lead to a mutation in the RNA. Mutation in the RNA, if it leads to a mutation in the codon, right? You're supposed to be GUC, then let's say it goes from, instead of GUC, you get GGC, right? You mutate this to a G. Instead of putting valine in the protein, at that amino acid, you're going to put glycine in it. But if you mutate the third one, it can be all 4. If that's mutated, you're still going to get valine in the protein. So it doesn't hurt you to mutate, to change that nucleotide. So that's what's built into the system. It's built in so you can change the last one in this one, the last one in the alanine, right? This one's a little bit different because it's only 2. But a lot of them that have 4, if they have 4 nucleotides, like glycine has 4, arginine has 4, what changes is the third one in all of them. The first 2 are all the same. That's a minor point. I don't even know why I'm mentioning it. So elongation is just the process of the tRNA coming in, reading the messenger RNA until you get to the stop codon. Right, so if you have 100 amino acids, you'd have 100 codons, and the last codon after that, or the 101 codon, would be a stop. Protein gets released. So, just like before, right, there's initiation, elongation, and termination for translation. The initiation complex, translation initiation complex, right, contains ribosomal RNA, messenger RNA, and transfer RNA. They all come together. And then elongation, peptidyl transferase is what makes the peptide bond between the amino acids. You're basically polymerizing amino acids, hopefully in the correct order. And then when you get to the stop codon, then you finish making the protein. Okay. So I'd like to put all this on here to emphasize, it's easy to mix up transcription and translation. They're spelled similarly. And it's nice to kind of probably remember DNA to RNA, messenger RNA to protein, or DNA to DNA for DNA replication. I mean, to do this justice, we'd actually spend a whole section on this, a whole section on this, or chapter, and then have a whole section on how to regulate all that. So we're going to get into the regulation today. So transcription and translation can occur at the same time in bacteria. So what we mean is, like on there, you say you have the DNA is at the top, right? And DNA or RNA polymerase copies a strand of it to make the gold strand here, right? That's messenger RNA. And then we've attached, right, this one ribosome attached to messenger RNA, say up here at the start, and it's already moved all the way to that position. And right behind it, there's another ribosome. So they call that a polyribosome, right? Meaning that there's many ribosomes on one strand of RNA. You're never really making one protein at a time. I mean, when you say you're going to express a gene and make a protein, you can be making anywhere from 50 to 100 to 1,000 to 10,000, 100,000 proteins. when your pancreas releases insulin into your bloodstream, it's probably releasing about 100,000 insulin proteins at once. Or if you inject somebody with so many units of insulin, there's a lot of protein in one little shot like that. You know, I don't know exact number, but it's a lot. But you need a lot of it, because once it gets into your blood, it dilutes. You're not going to use every single one. So, now I kind of put this on the board early on, right when we first started talking about the promoter. It's the on-off switch. So with bacteria, they have an organization called an operon. So let's say here, this is a piece of DNA. And let's say at the beginning here we have the promoter, right? We said that's the on-off switch. Let's say this part is the promoter. And then we have 1, 2, 3 genes. And we'd say those 3 genes are under the control of this promoter. In bacteria, that's the way. Now, if it's eukaryote, you know, like us, we always have 1 promoter, 1 gene. I think that's pretty much all the time. What bacteria will do is have, say, 3 genes, right? And you make a messenger RNA, and then you make protein number 1, protein number 2, and protein number 3. So when the reason they group these 3 proteins together is because they all serve the same function. So in this example, these are 3 enzymes, and they're used to ferment lactose. Right? If a bacteria wants to ferment lactose, there are 3 genes that are needed to do that. It's not just 1. So what does we mean by ferment lactose? Right? They are converting lactose. They're using it to make ATP in cell respiration. They have to convert to glucose to do that. But without these 3 enzymes, they can't, right? They are unable. So if you do the lab and if we say, if lactose is fermented, the colony becomes pink. That it also means that the bacteria has these 3 enzymes. They're able to ferment lactose, make ATP, or eat the, it's basically they're eating the lactose. To do that, they would have to turn this promoter on, make the messenger RNA, and convert the messenger RNA into protein. Express the proteins. What if they're not? What if lactose is not present? Then they don't need these 3 proteins because there's no use for it. Nature's pretty smart. It knows, it'd be wasteful to make a protein that you don't need. It takes a lot of energy for the bacteria to make it. So what they do is there's a repressor that sticks to the promoter. As long as the repressor's here, it represses transcription. It blocks RNA polymerase is another way of thinking of it. RNA polymerase can't come to the promoter and make a copy of the DNA. No protein. Turned off. Well, what if you put the bacteria on a plate that contains lactose? Well, nature has figured out what to do, right? It takes the lactose, it gets into the bacteria, it binds to the repressor. By binding to the repressor, it kicks the repressor off. So it was turned off, the promoter, by the repressor being present. When the repressor gets bounced off, the promoter gets turned on, and you express the 3 genes that are needed to ferment the lactose that's present. That's what we mean when we say lactose induces transcription of the 3 genes needed to ferment it or metabolize it, however you want to say it. All right, we've got 20,000 genes, and at one time we're not expressing them. In some of those 20,000 genes, they're never expressed in some cells. Oh no, that's not true. There's always a few. So, now, this is a little bit different. Instead of fermenting something and breaking it down, now we're saying we've got 3 genes that are needed to make arginine, right? If we want to make arginine, which is an amino acid needed to make proteins, in other words, it always needs to be available. But what if we have more than enough arginine? Or the bacteria does. Then if these 3 genes are needed to make it, one way of stop making it is not to make the proteins that are used to make the arginine. So what happens is when arginine accumulates, and there's more than enough of it, it binds to the repressor. It's a different one this time than the other one. It looks a lot like it, but it wouldn't be the same repressor. And it causes the repressor to stick to the protein, or to the promoter. If the repressor sticks to the promoter, in this case, just like in the other case, right, the promoter's turned off, and you don't make the enzymes because you don't want to make arginine. So it's the opposite of before. Before you wanted to induce something, you wanted to induce enzymes to ferment something. Here, you only want to induce these proteins if you need to make arginine. If you've got plenty of it, then you don't need to make it. So when the arginine levels go too low, then the arginine is no longer attaches to the repressor and it falls off, and you make these enzymes that make arginine. It just happen to be 3 enzymes that were needed, and arginine's made. So arginine, kind of like the opposite, can block transcription of genes needed to make it. So this is more or less like a little biochemical pathway. You know, enzyme 1 makes something, enzyme 2 makes something, and then enzyme 3 makes arginine. You don't need to know that biochemical pathway, but I'm just sort of talking about that. So just to wrap that up, right, enzyme induction is when the expression of enzymes are, you get expression of enzymes in response to the presence of a specific substrate. That was the lactose example. Enzyme repression is you block expression of enzymes in response to a signal. In this case, it was arginine, right? The signal means